Welcome back. It's chapter 17, part 3. This is a very short video on mutations. And so we're thinking about transcription and translation in chapter 17. And, excuse me, let's get started on the last component of chapter 17. I will bust over to my slides. I don't know which ones they are. Here we are. Okay, so we've covered transcription and translation. Transcription is when we start with a piece of DNA, a gene, and copy it into a single-stranded RNA molecule called a messenger RNA molecule. This happens in the nucleus. Of course, that's where the DNA is, right? And then this RNA molecule is modified, it's processed, and it matures, and then it heads out into the cytosol, or the cytoplasm, where it is translated. And in translation, ribosomes bind to the mRNA molecule. tRNA molecules that recognize the codons are bringing in appropriate amino acids. We're building a chain of amino acids. And we read in translation the codon code. The mRNA molecule is read until you arrive at a stop codon, and then all the parts are released, and we get a cute little polypeptide chain, right? Okay. Sometimes mistakes happen. Point mutations are small mutations that can occur, or small changes in the nucleotide sequence. Okay? And when you have a point mutations can happen during DNA replication. Oops, a little error was made when I replicated my DNA, and now I have a small change in my nucleotide sequence. Or, and then that change stays with us, stays in the DNA, in all of the cells' descendants. So a point mutation is simply a small mutation involving a small number of nucleotides in the DNA, in the original DNA sequence that's inside the nucleus of the cell, right? In here. When you've got a point mutation, that can, but isn't always, devastating to a cell. It can be problematic because it can lead to the transcription and translation of a protein that doesn't work. And there are different kinds of mutations that are worth taking a look at. The consequences of mutations are also worth thinking about. I'd like you to read this section on your own. That's why this video is so short, but I wanted to point out a couple of things. Here's an example of a point mutation that can occur in a gene that codes for, the gene is actually called the beta-globin DNA gene, or the beta-globin gene. This gene codes for a piece of hemoglobin, the beta component of hemoglobin. Now you have to step back and remember what hemoglobin is. Hemoglobin is a protein that's super abundant in red blood cells. And hemoglobin is made up of kind of 4 parts, 2 alpha subunits and 2 beta subunits that are put together to make this complex protein. And hemoglobin does the job of transferring oxygen in our cells, right? And also it helps transfer carbon dioxide. Our red blood cells are just, they're usually cute little donuts like this one, and they're just packed with hemoglobin, transporting oxygen. Okay. Now, people who have sickle cell disease sometimes have their red blood cells do this number, take on a sickled shape, and that's because there's a problem with the hemoglobin inside those red blood cells. And here's the problem. There's a point mutation in one of their genes, the beta-globin gene, okay? And we've got the wild type, or this is kind of the normal version of the gene. And of course, we're not showing the whole gene. We're only showing 3 nucleotides in this whole gene. And it turns out that in the wild version, this gene is transcribed into an mRNA molecule, and it's translated into a series of amino acids. Okay, so these purple blobs here represent, each of these represents a different amino acid. And the normal hemoglobin has GLU at this particular amino acid. Okay? In people who have sickle cell disease, their beta globin gene has a point mutation in it. Just one nucleotide has a mistake. Instead of a T, there's an A at this location. What that does is it changes the mRNA codon from GAG to GUG. That, during translation, is translated into valine instead of GLU. Okay? So there's one amino acid that's different in this whole molecule, in this whole polypeptide that's used in hemoglobin. Just one tiny amino acid change. That leads to all of the problems. So what we've got here is a picture of normal hemoglobin. It's made up of two different parts. So we've got an alpha protein, an alpha polypeptide, a beta polypeptide, and a beta polypeptide. And it turns out that in people with sickle cell disease, their beta hemoglobin component, this polypeptide, has a slightly different shape because, guys, of the single change in one amino acid due to a point mutation in their DNA. Okay, what does that mean? Well, most of the time, this hemoglobin in people with sickle cell works pretty good. It carries oxygen and carbon dioxide pretty well. But occasionally, under certain periods of duress, like under low oxygen conditions, or when a person is fighting an infection, or maybe other situations when the person's really dehydrated, their hemoglobin, instead of kind of just keeping separate, forms a long chain, and that chain causes the whole red blood cell to become sickle shaped. And if you have sickled red blood cells, you can have all sorts of problems. These sickled red blood cells get caught in your capillaries, and they cause pain in your extremities because you can't get blood flow to the extremities. Sickle cell shape can lead to an increased risk of having like a clot or a clot and that causes an increased risk of stroke. Lots of issues with having a sickled red blood cell. And another situation would be your immune system sees that sickled red blood cell and goes, that's a mess, I got to get rid of that. And so they send those sickled cells off to the spleen to be destroyed. And pretty soon you don't have enough red blood cells. And you don't have enough oxygen carrying capacity, so you have anemia. Lots of symptoms associated with sickle cell. And the key is, all of these symptoms occur because of one tiny point mutation in a single gene in your DNA. Crazy. That leads to, after transcription and translation, a polypeptide chain that has one amino acid difference. And with one amino acid different, it leads to a different shape in your part of your hemoglobin molecule, and that can lead to the trouble. So sometimes a point mutation can have devastating effects. Sometimes point mutations don't have any effect. And this slide is something you're going to have to spend some time looking at. But basically, in this slide, we have a picture of the DNA in blue, a picture of the mRNA in red, and a picture of the polypeptide chain, or the sequence of amino acids, in purple. All right? And the DNA is virtually the same in all of these drawings, but there might be small changes. All right? So there are different kinds of small or point mutations that can happen to DNA. So there can be a substitution. Oops, instead of, and here's our original DNA molecule, okay? So now these represent, on the bottom here, these represent all of the different types of mutations that can occur and what that means for the resulting protein that's made. So up here is kind of the normal version. We would call that the wild type version. That's what the language we use, the wild type version of the DNA. And it leads to a polypeptide chain that's 4 amino acids long. What if we substitute, have a tiny substitution, okay, an A instead of a G? What's going to happen? Well, in this case, there's no effect, all right? This is a silent mutation. But if you change if you make a mistake in a different nucleotide, there's a mutation in a different nucleotide, you could have a change in the amino acid sequence that produced. Oh, it looks like our 4th amino acid here is different. It's called a missense mutation. Or this could be even worse. What if you have a small, just a single nucleotide mutation, a substitution mutation, if you will, and that leads to the production of a stop codon early on, and then you don't even finish making your protein. It just stops, and so we call that a nonsense mutation, or that the consequences of this is a nonsense consequence. You got a real problem then. You didn't even make your polypeptide. You just stopped short. That could be problematic. In addition to substitutions, you can get insertions or deletions, and these are really devastating because what that does is it frame shifts the reading of your mRNA molecule. If you add an extra nucleotide in place, that messes up the whole rest of the reading of this mRNA molecule. Everything has to be shifted over, and that can lead to all sorts of problems. Okay, here's a it looks like we're deletion. Here we've got a deletion. Again, lots of problems. Okay, when you delete or insert a nucleotide, that changes the reading of the entire mRNA. From that point on, there's going to be mistakes. So those are kind of devastating point mutations. Okay, that is the end of chapter 17. There are some good test your understanding questions that are worth checking out. And